is exciting. I've missed my viewers so much. It's Major Bumsaw here. Neil isn't here at the moment, and to be honest with you, I think this video would be a lot better if I was presenting it instead of him. Don't you think so? You know what I'm saying?
Oh, I'm really excited about today's episode. It's going to be great. What's that? There's someone there. Boy, wake up.
What did you do that for?
Major Bumsaw.
Oh, hello, Benito. How are you?
What the hell are you doing here?
Well, you know, just taking advantage of the moment. I mean, I haven't been in a bloody episode for 2 years. What have you got to say to that?
Well, actually there's a reason for that, because this is actually a proper intellectual channel now, you see, so it's quite...
No. Well, that's where you're wrong, Benito. I have some feedback from the viewers, and they clearly say that I'm the one to be presenting this show.
What?
So, in your face with that, bitch.
Whoa, yeah. Anyway, we need to look into that, but at the end of the day, the final decision is mine, so I will take that out. Hello everyone, and welcome to DNA Expression Implants. It's going to be great. It's going to be... Oh my god! It's annoying.
Good God, that was very rude, Benito.
Well, I don't care, because you need to go now, because I can't cope with you anymore, so...
There's nothing you can stop me.
Yes, there is. If I just do this. Maybe.
No.
Yeah.
No, don't do that. No,
Yeah.
no,
Yeah. I could do something else as well. Yeah. Sorry about that everyone, and welcome! It's the 8th instalment of my molecular methodology series. This is crazy! This is going viral! Well, actually, it's going botanical because we're talking about expression of genes in plants. Do you get what I did there? Okay, I'll move on. And get on with it I should, because when I started preparing for this video, I started off thinking, well, I did fungi in animals, okay, so this should be fine. But then, I forgot that there's a whole ton more to learn with plants, so this is going to be pretty hectic. This could be the most intense video you have ever watched in your life ever. So feel free to pause it whenever you like when you're not getting what I'm talking about. That means this video is probably going to be split up into 2, because otherwise it would just be too much for me and your head would explode. I'm serious. Now, the good thing about plants compared to animals is that they don't have a germline. Remember last time we talked about the germline of animals? Well, all the gamete producing organs in plants all derive from somatic cells. So you'll get the shoots which are somatic and they'll produce branches and you'll get flowers at the end. And remember, it's the stamens and the carpels within those flowers that produce the gamete, so they are the germline, but they're all derived from the soma, the somatic cells. Another thing I should point out that this video will focus primarily on dicotyledonous plants, so dicots. We'll move on to monocots in probably the second part of the video, but basically all I'm going to say in this video is about dicots. Now the good thing about plants is that if you take a segment of leaf tissue, for example, and place it on a nutrient medium with hormones, it's 2 particularly important hormones, that's auxin and cytokinin, those leaf cells will divide and eventually should give you a perfectly new plant. Well, that's very useful when it comes to making transgenic plants, which is what we're going to be talking about. Now, if you have the auxin and the cytokinin in equal amounts, then you'll get what is called a callus, just a plate of undifferentiated cells that aren't really doing anything at all, it's just a collection of plant cells. However, if there's more auxin in your nutrient medium than your cytokinin, then you'll get roots developing. So these cells will differentiate to produce roots. Likewise, if you have more cytokinin than auxin, you'll get shoots developing. So all you need to do is to introduce the foreign DNA that you want to insert into a plant tissue culture, and then from that you can develop a whole new plant, a whole transgenic plant. But obviously, it's a lot more complicated than that. Now, in Dicots, transgenic plants are made using the agrobacterium called Agrobacterium tuberfaciens. Now, we're going to take a step back for a moment and look and see what Agrobacterium tuberfaciens does in nature. Okay, that's an actual bit of biology in these revision videos, isn't it great? And then we're going to look at how we can apply that to making transgenic plants. Now, Agrobacterium tuberfaciens in nature causes something called crown gall disease. So that's to say, when this agrobacterium infects a plant, it increases cell division, cell growth, so cells will continuously divide to produce this gall, this tumour, this cancerous tissue. Now they're called crown galls because they produce these galls at the crown of a plant. Now what's the crown of a plant? Well, it's the point where the plant meets the surface of the soil, and it gets into the plant through little cuts in the shoot. And the crown is particularly vulnerable to that because, I'm going to do an impression of a plant here. This is the ground. My bicep is the soil surface. My forearm is the plant, and as the plant goes blown about in the wind like that, it'll probably produce little cuts around the crown of the plant here, and that's how the bacteria gets in. Now, if you extract some of this gall tissue and put it on a medium without any plant hormones, then the gall will carry on dividing. They'll carry on proliferating. So this is called hormone-independent growth. But how is it doing it? Well, those galls must contain a gene which allows them to produce their own plant hormones, so auxin or cytokinin. This is because a part of the agrobacterium genome has become incorporated into the plant chromosome and therefore getting expressed. We'll move on to the nitty-gritty of that in a second. Another thing that the gall produces are opines. Opines are condensation products, so products that have come out of a condensation reaction, which produces water, of normal plant metabolites. So, for example, the opine called octopine is made up of arginine and pyruvate that have reacted together in a condensation reaction and have been covalently bonded together, so they're now one molecule. Now, normal plant cells don't produce opine, so the gene for opine production has also been incorporated in the plant genome. And these opines are important because they act as a food source, if you like, for the agrobacterium themselves. It's effectively making its own food. It's what's giving the agrobacterium more energy so it can colonise more plant cells and infect them. So, how is all this genetic material incorporated into the plant genome? Well, it's done using this beautiful thing, the TI plasmid. TI, not that famous American rapper, unfortunately. Well, unfortunately, he's pretty crap, isn't he? But no, it is this. Now, what have we got here? Well, the bit of DNA that's incorporated into the plant genome is called tDNA, transfer DNA. And I've labelled that here. I don't know whether you can see that very well. And it's within that transfer DNA that you'll find your octopine gene, so I've just said it somewhere there, for example, it could be anywhere, and you've also got genes for auxin and cytokinin. So that's what allows the cancerous cells to proliferate, even in the absence of external hormones. So that's very important. And this tDNA is surrounded by these 25 bases on either end. And these are recognised by an enzyme, so when transcription occurs of this tDNA, that enzyme will cut there, and this tDNA can slip into the nucleus of the plant cell and be incorporated within a chromosome. Now, I haven't talked about all the other things on there. Well, we've got our octopine catabolism gene for digesting octopine in this case. It could be any opine, we've just chosen octopine as an example. We've also got a virulence gene. Now the virulence gene acts as a first point of call, if you like, because what it does, chemicals released by the plant at a wound site activate the protein produced by this gene and allow it
Right,
to
I'm going to take over here because I
transcribe
actually
the
made a bit of a mistake
tDNA,
during recording, silly me. So what the
segment,
activated product of the
which eventually
virulence
gets
gene
incorporated
does
into the genome.
is that it detects the 25 bases on either side of the
So, chemicals
tDNA,
produced by the
and it
plant at the
cuts
site
it,
of a wound
forming a linearized
activate virulence
tDNA
gene.
molecule.
Virulence gene tDNA goes into plant cell, into nucleus, integrated into the chromosome at a random place, and then starts transcribing the auxin, the cytokinin, and the octopine. All done and dusted. Oh, and to ease the transfer between the tDNA from the agrobacterium into the plant, it's also surrounded in a nice protein envelope, so it's more easily transported between the two types of cell. Now what's interesting is that all the species within the genus Agrobacterium are defined by the type of TI plasmid they carry. So depending on what opine that this TI plasmid produces, it depends what species it is. So how do you effectively transport that tDNA from the agrobacterium into the plant cell? What you do is you use a binary vector system. And this is it, the binary vector system. Now this allows you to incorporate your foreign DNA that you want to insert into the tDNA, which is eventually going to be integrated into the chromosome. And, as the name suggests, you use not one, but two vectors. Okay, so here's number one and here's number two. Now what's going on? Now, number one doesn't seem to have a lot going on it. That's because it doesn't really. This is what we call the disarmed plasmid. What it has is an origin of replication, very useful. It also contains the virulence operon that we've been talking about. It contains no tDNA whatsoever. There's been a simple mutation occurred in this plasmid, which means that no tDNA is produced. On the other one, however, is where the real stuff happens. Now this one is confusingly called the binary tDNA plasmid. We have our origin of replication. It has a broad host range because we're going to be using this vector in E. coli and Agrobacterium, so it needs to be able to replicate in both organisms. So that's why we use a broad host range origin of replication. This L and R just signifies the left and right side of this tDNA. So between the left and right we have the tDNA, and that's what's going to get sent into the plant. Now we've got two things here. We've got the selectable marker, and obviously we've got our foreign gene. What? Now you might be thinking, what about the genes for the opines and the genes for auxin/cytokinin? Well, do we really need them for this particular procedure? No, we don't really. And what we want is to get rid of as much garbage as possible. In fact, if we did include them, then it will be more difficult to get this plant to differentiate into a completely new plant. You just get masses and masses of cancerous tissue everywhere. So it's important that we cut them out. And that means a greater proportion of this plasmid is the foreign gene that you've inserted, so it's easier to work with. So how do you insert these genes, well, for the foreign gene and the selectable marker, into the plasmid? Well, this is similar to stuff we've done before, but it's just been given a different name. What we need is a plant expression cassette. Okay, and this is shown quite neatly by this diagram here. Now, this is in the case of producing a selectable marker. And the selectable marker which we're using in this experiment is for kanamycin resistance. And that's symbolised by the MPT2 gene here. Don't worry about what NOS stands for, that doesn't really mean anything. Now the kanamycin resistance gene is very, very useful because it inhibits protein synthesis in bacteria. And through this we can use this as a selectable marker in bacteria and also in plants. Why also in plants? Well, if we stick in our evolutionary biology hats on here, then we should know from the endosymbiont theory that the chloroplasts within eukaryotic plant cells are actually prokaryotes. They're bacteria. They were once cyanobacteria that were engulfed by a eukaryotic cell. So synthesis of proteins produced by the DNA in the chloroplast will be inhibited if we introduce kanamycin. So if we have a kanamycin resistance gene as a selectable marker, that's very useful because it allows us to select for transformants in bacteria and in plants. So what you do, you've got a gene between a promoter and a terminator. All you need to do is to cut that out with restriction enzymes and stick in the new gene that you want. Downstream of the promoter, and so it links with the terminator. Remember, the terminator produces the polyadenylation signal, which increases the stability of the mRNA transcript. So what you end up with is a chimeric gene. It contains DNA from two different sources, and it will be translated because the transcriptional proteins bind to the promoter and transcribe all of this into mRNA, which can then be translated. Now, the original binary expression vector to use for this process was the one and only PBin19 binary vector. Oh, and what a good binary vector it was. Obviously, P stands for plasmid, bin stands for binary, and 19 is because it contains sections from the PUC19 plasmid. Now, what's special about this particular vector is that your multiple cloning site within your tDNA, between your right border and the left border, the multiple cloning site is downstream of the LAC-Z alpha promoter. Ooh! Sorry, I overreacted there. Now, we've spent countless times talking about the LAC-Z promoter, so if we insert it downstream here, we can do a simple screen with Gall. If the solution turns blue, we've got non-recombinance. If it turns white, we've got recombinance. Another couple of things which are different here is the RK2 origin of replication. That is just an example of an origin of replication with a broad host range, so we can use it in E. coli and we can also use it in Agrobacterium tuberfaciens. This APH1 gene here is another kanamycin resistance gene. So I think I've given you all the information you need now for me to go through the entire process in one go. Are we ready for this? Let's get going! So, first of all, you've got your two vectors. You've got your disarmed plasmid and your binary tDNA plasmid, which we've been talking about to death. In this case, we're going to be talking about the PBin19 binary vector. Now, we leave the disarmed plasmid as it is. We don't need to mess with that. What we're going to do first is introduce this plasmid into E. coli. Allow it to clone and replicate this plasmid many times, and we're then going to select for transformants used by introducing kanamycin. Those which have taken up the plasmid will have our kanamycin resistance gene, so will survive. Ones that don't will die. We can also do a simple screen with Gall. Non-recombinants will be blue, recombinants will be white. So all the colonies which are white are the ones we want. So we extract the plasmids from those white E. coli colonies. And then we're going to try and get them into agrobacterial cells. And we do that through electroporation. Now, I mentioned electroporation very, very briefly in the first ever video of this series. What we do is basically insert an electrical voltage which creates pores in the cell membrane and the cell wall. And it's through these pores that our plasmid will slip into and fall nicely in place alongside our disarmed plasmid here. And then what we do is what we talked about before, the leaf disc transformation event. We take our agrobacterium, we take a circle, let's say, of plant leaf tissue. That means the virulence gene of your disarmed plasmid will be activated. It will then find the 25 special bases just before the start and end of the tDNA fragment, and it will snip them. So you've got your linearized tDNA. That will then slip and move in to the plant cells, encapsulated in a protein coat. It will move into the nucleus and get incorporated into the genome of the plant cell. So once the agrobacterium has done its job, we don't need it anymore, so we kill it off with some antibiotic. We use kanamycin to select for those plant cells that were transformed, and then we introduce hormones into our medium, the auxin and the cytokinin, which produces this callus, this undifferentiated plant tissue. Now we then increase the cytokinin level. Now remember, as I said earlier, more cytokinin to auxin, that means shoots will be produced. So this shoot can be cut, introduced to a root-inducing medium, i.e. contains a lot of auxin, and that will produce the roots. You've got your shoots and your roots, you've basically got a plant. So there we have it, a transgenic plant. Every cell in that plant contains the foreign gene that we inserted in the original transformation event, because they all originated from that one cell that we transformed. However, it's not homozygous, is it? Just like how we talked about in the last video, this is a hemizygous plant, because the tDNA has only been incorporated in one of the homologous pair of chromosomes. Once again, we're looking for homozygous, and to do that, it's relatively easier compared to animals, because plants have male and female parts. So what we can do is self-fertilise. So when you cross two hemizygotes, you'll get your classic Mendelian 3-to-1 ratio. 3 to non-homozygotes to 1 homozygote. So those homozygote seeds will be grown and we can produce a fully transgenic plant. But remember, it's a 1 to 2 to 1 ratio, so we'll also have one completely wild type, which is useless to us, but 2 hemizygotes. So how do you distinguish between the wild type progeny and the hemizygotes? Well, it's simple. You just grow those seeds on the medium containing kanamycin. If they die, they're homozygotes. If they survive, then they're hemizygotes, because they have this selectable marker incorporated in their genome. And then we can then crossbreed them to get more 3-to-1 ratios and more homozygotes, which are ones that we really want, because they're the ones that properly express the foreign gene that we've inserted. Well, if you're feeling emotionally drained after that video, then I don't blame you, because I am really. So we're going to leave it there for a moment, but I'll be back very soon talking about how we transform not dicots, but monocots. Which, trust me on this, is simpler, and we use DNA guns and stuff. It's going to be awesome! Wah! And, more importantly, this is going to be the last molecular video which I do before I move on to something, frankly, a lot more interesting, and that's going to be evolution! Wah-hey! But for the moment, I think I need a little break, so if you'll excuse me. Or